main challenge with AAV system, as I point out, the AAV is very small virus, and it's been challenged to develop this system, and so far there is no such system exists. So closest to this would be studies by Kevin Young and Charlie Gershbach at Duke University, and they utilize so-called dual CRISPR-Cas for epigenome editing, meaning two vectors, one expresses guide RNA, a crucial component of CRISPR-Cas, and second expresses Cas9, small Cas9. This in those study they utilize a strain from Staphylococcus aureus, SA, so-called SA Cas9, combined with a crab repressor. So two vectors delivered into systemically or into CNS, depend on disease designation, what they're trying to achieve, and they demonstrate very good results and successful correction of disease relevant phenotypes. However, again, the main disadvantage of so-called dual approach those studies utilize is that you need to, first of all, you need higher load of AAV because it's two vectors. One, again, for delivering guidance, and second for delivering Cas9 / a repressor. So you need double dosage, which again, it's very safe platform, but it has relative shortcomings with immunoreaction, and obviously it would be preferable to lower the viral dose. And second, the efficiency in those studies weren't so great, and it's from obvious reason. You really need that two vectors, two genomes will meet in single cell to generate this effect, which again will require higher dose, and from obvious reason it's not achievable at very high percentage rate. So for this reason, I believe that our development of all-in-one, when you deliver all components in from single AAV particle, is beneficial and preferable approach. We just submitted publication, which is currently under review in molecular therapy with all this development, and we also reported a week or so ago. It just came from a AAC conference in St. Louis, Missouri, where we reported those findings as well. So I do believe it has it's very attractive therapeutically wise approach, and yes, we're excited to move this system through the small animal study, large animal study, and all the way to with ultimate goal to apply it in clinical trial. And this is very crucial. This is very important. What will be the delivery schedule of those drugs? And so absolutely preferable and sort of requirement that it will be used one time. So you don't want to apply those tools over and over, especially when you go to CNS. So we demonstrated that this actually could be possible because adeno-associated vectors are a transient system. So when cells divide, AAV genome cannot be sustainable. They will be diluted out with cell division. They don't have origin of replication. They cannot survive in dividing cells, right? So we demonstrated actually that this epigenome editing is permanent in its nature. So, for example, we use this AAV to transduce high-pace dividing cells, and we show that even after 3 weeks, when all the genomes of AAV are gone because of cell division, the effect of repression persists. And that's very important. And as I pointed out earlier, that's in debate in the field whether those epigenome modification created by this approach will be inherited and will be permanently expressed their effects. Now, for CNS delivery, it's less, it's possibly less sort of important because CNS, as we know, mostly comprised from post-mitotic cells, not non-dividing cells. But still, it's very, very encouraging that regardless of cell division, regardless of whether cells are dividing or not dividing, the effect is inherited and persists over cell division. And obviously, we didn't test whether it will be persist in such a way in CNS, but assumption if in dividing cells we see effect, it likely, very likely, we'll see this mode of effect in neurons or in CNS. So that's another important point.